Hello everyone, how are you? I hope you are doing well. In order to help students, especially exit exam takers in near future, I have prepared question teams in biological science, particularly in biotechnology. As you have seen video number one in my YouTube channel, many questions are uploaded for the course genetic engineering. Based on that, many students asked me to discuss upon those questions. As a result, I need to have it in this video number 2. I wish you good lesson in the discussion. Before we go to the next page, please subscribe and like the video to get the next new video. Question number 1. What lessons can someone in the biotechnology department with a BSc program expect more from the genetic engineering class? In these questions, lessons and genetic engineering class. Lessons and genetic engineering class are the indicators or keywords for these questions. What lessons do you expect in the genetic engineering class? This question is simply to remind you the course description of genetic engineering. At the beginning of your class, your course teacher might introduce you about the course description. He or she told you that genetic engineering taught about gene manipulation tools, including vector, enzyme, host, marker, laboratory skills. Those are some of the gene manipulation tools in genetic engineering. In addition, the course genetic engineering taught us about gene library construction systems, that is, a genomic library and cDNA library. Then uh, gene amplification techniques, also the other important topics in genetic engineering class. These gene manipulation techniques include the molecular cloning, which is the in vivo amplification technique of gene, and PCR, which is the in vitro amplification technique of a given gene. Recombinant technology, also the main topics in the course genetic engineering. Gene transformation systems across organisms. This may be biological systems, the physical system, and also chemical method of gene transformation. Then the transgenic organism, transgenic plant, transgenic animal, and transgenic microorganism. The application of those transgenic organisms over different applications. The application of genetic engineering in agriculture, in medicine, environment, and industry. This and other disciplines or subtopics we'll discuss in the course genetic engineering. So in this question, which one of the following or which type of lessons are expected in genetic engineering class? Choice A says nucleic acid metabolism. This is not that much appropriate for genetic engineering class. B, cloning vectors and cloning hosts. Yes, this keep correct. C, micropropagation of plant. This is not appropriate for the course genetic engineering. This is not a direct lesson for genetic engineering. D, molecular absorption and metabolism. This is not a direct class for the course genetic engineering. So from those given alternatives, B is the correct answer for question number 1. Question number 2. As a biological science student, why do you take genetic engineering course? Why do you take is the main keyword here for this question. Why do you take the course genetic engineering? This is asked at the reason why we're going to learn the genetic engineering course. This is simply to know, to identify, and to understand different topics and subtopics that we discussed in question number 1. So alternative A says to identify various applications of recombinant technology. Yes, recombinant technology is the main topic in the main lesson in genetic engineering class. Therefore, we're going to understand recombinant DNA. What is recombinant DNA? What are the tools for recombinant DNA? And the mechanism to form recombinant DNA. We're going to understand, identify the tools, and we're going to know the mechanisms. B, to know the basic principle of gene manipulation. Yes, how to manipulate gene. What are gene manipulation tools, and how to use those genes for different applications through this principle. So B also correct. C, to understand cDNA synthesis. This is one of an important principle to construct a genetic library and also used to different diagnosis. So cDNA synthesis is also another important issue in genetic engineering. So it is correct. All of the above are correct for the reason that we're going to learn the course genetic engineering. Question number 3. Among various modular categories, in which category is genetic engineering classified? In this question, the keyword is modular category of genetic engineering. In which modular category is genetic engineering classified? So therefore, modular category for genetic engineering is the keyword. Based on these questions, this is simply notify you about a biotechnology unit or streams. There are 5 biotechnology units nowadays that are agricultural biotechnology, including the course like plant biotechnology, animal biotechnology, plant breeding, animal breeding, plant cell and tissue culture, plant physiology, animal physiology also. The other biotechnology unit is medical biotechnology, consists like courses pharmaceutical biotechnology, diagnosis technology, immunology, and immunotechnology courses. The other is environmental biotechnology. In this stream, there are courses biosafety, biodiversity conservation and management, aquatic biotechnology, environmental biotechnology, and others. The other is industrial or microbial biotechnology. In this stream, we're going to learn about enzyme technology, food biotechnology, microbial biotechnology, and others. The other stream is genomic science and bioinformatics. In these streams or category, they are genetic engineering course and bioinformatics. Genetic engineering and bioinformatics is classified under genomic science and bioinformatics unit. Besides, there are other basic biotechnology courses like molecular biology, biochemistry, biological laboratory techniques. So, based on this description, the course genetic engineering is classified under which of the following alternatives? A, agricultural biotechnology, it is not appropriate. Industrial biotechnology, it is not. The genomic science, genomics and bioinformatics, yes, this is keep correct. Medical biotechnology, it is not. Environmental biotechnology. This is simply the classification of courses under different modular categories. It doesn't mean the course genetic engineering doesn't applicable for those other streams. It is very applicable. So this is a modular classification or category. So choice C is the correct answer for question number 3. Question number 4. Which biomolecule is responsible for the transmission of hereditary information from one generation to the next? Which biomolecule is responsible for the transmission of hereditary information? The keyword is hereditary information. Here also biomolecule. All those alternatives are the biomolecules. So this is not it is not used as a keyword. So the best keyword is hereditary information. All those biomolecules have their own unique function within the organism. There are four biomolecules in the organism. They are biomolecule, they are organic molecules. Those molecules have their own functions. Those biomolecules are protein, which is used for body support. The other is lipid, which is used for long-term energy storage and structural units. For example, cell membrane is composed from lipid. The other is the nucleic acid, which is used for amino acid synthesis. This amino acid for protein, that protein going to express the phenotype that we're going to look in the physical environment. The other function of nucleic acid is the hereditary information storage. It is the center of hereditary information. Hereditary information is the biological information that transfers from parent to progeny, from the generation to the next generation. The other biomolecule is carbohydrate, which is used to store immediate energy. For example, starch for animal energy storage and glycogen for glycogen for animals and starch for plant energy storage. And also this carbohydrate used for structural unit. For example, the cell is composed from the cellulose. So based on this description, the biomolecule which is responsible for hereditary information is nucleic acid. Again, C is the correct answer for question number 4. Question number 5. The biomolecule that controls the discrete hereditary characteristics in all organisms except some virus is blank. In this question, the key word is biomolecule. Actually, all those alternatives are the biomolecule. Therefore, biomolecule is common for all, but discrete hereditary characteristics is an important keyword. Hereditary characteristics in all organisms except some viruses. Those are the indicators for the specific answer from the given alternatives. In this question, we can remember that there are two types of nucleic acids. Those are DNA, that is deoxyribonucleic acid, and RNA, which is the ribonucleic acid. DNA is found in all organisms except some viruses because some viruses have RNA and called RNA viruses. Therefore, all organisms except some viruses consist a DNA. Therefore, from those given alternatives, which one is best or specific answer for the given questions for question number 5? Alternative A says nucleic acid. This nucleic acid is general, which consists DNA and RNA. Therefore, it is general rather being specific to control the hereditary characteristics in all organisms except some viruses. B. Ribonucleic acid. This is only in some viruses, only with some viruses. C-deoxyribonucleic acid. This is DNA, the DNA which found in all organisms except in some viruses and control all hereditary information or traits of that organisms. The nucleoside. This is not a macromolecule, rather the monomer for nucleotide. A monomer for nucleotide. This nucleotide with other nucleotide is a nucleic acid. This nucleic acid is either DNA or RNA. Therefore, the correct answer for question number 5 is C, deoxyribonucleic acid. Question number 6. The group of nucleotide sequence, that is not found in prokaryotic organism is blank. This question, nucleotide sequence, all those are nucleotide sequences. They consist different nucleotide sequences, but the one which is not found in prokaryotic, is the keyword for this question number 6. In this question, simply to restate about the type of cells. There are two types of cells. These are eukaryotic and prokaryotic, which is based on the presence and the absence of the nuclear and organelle membranes. Those cells which have the organelle and nucleus membrane are the eukaryotic, and those which have not nucleus and organelle membrane are the prokaryotics. Prokaryotic cells. Those cells have the nucleotide sequences in their cell organelles and in their chromosomes. They have nucleotide sequences. The nucleotide sequence consists the intron and exons. The introns are the non-coding sequence, while the exon is the coding sequence. But intron is not found in prokaryotic. Therefore, the correct answer for question number 6 is what? A, promoter. This one is a nucleotide sequence which is to initiate transcription. Transcription is take place both in prokaryotic and eukaryotic. Intron, a nucleotide sequence with a non-coding region of that sequence, the non-coding sequence of the genome or a nucleotide sequence. This one is non-coding. Exon is coding sequence of a genome. Coding sequence of the genome. Enhancer, also a nucleotide sequence which facilitate transcription. So based on our statement, the introns are not found in prokaryotic. So intron is, or alternative B is correct for question number 6. Question number 7. Which of the following compositions is correct for the formation of nucleotide? For this question, composition of nucleotide are the keywords for this question. Which composition, which atom, or which molecule is used to form a nucleotide? Here, nucleotides are the monomer of, or a building block of, nucleic acid. They are a monomer or building block of the nucleic acid. There are nitrogenous bases, which is made the sequence of that nucleic acid or nucleotide, and ribose sugar and phosphate that formed the strand of nucleic acid. They are the side chains in the double helix DNA. So the nucleotides are composed from three important components. Those are nitrogenous base, the other is the sugar, the other is the phosphate. Sugar and phosphate. Nitrogenous base form the sequence or the ladder in the case of double helix DNA, and sugar and phosphate made the side chains. So the correct composition of a nucleotide in this alternative is which? Nucleotide is nitrogenous base and phosphate group. No, this is not the correct combinations. B, nucleotide is ribose sugar and phosphate group. No, this is not the correct combinations. Nucleotide is nitrogenous base ribose sugar. No, this is not nucleotide, rather nucleoside. This is a nucleoside. Nucleoside is a monomer for that nucleotide. D, nucleotide is nitrogenous base plus ribose sugar plus phosphate group. Therefore, this consists nitrogenous base, ribose sugar, and phosphate group. So the correct composition of nucleotide is those three components. So D is a correct answer for question number 7. Question number 8. What factor or factors will influence the expression of a particular phenotype in organisms? That indicates the factor that influence the expression of one's phenotype. Here there are keywords. We're going to select factors that influence the expression of phenotype. Phenotype is the physical appearance of an organism or the physical outlook of an organism that we're going to observe in the physical environment. So there are three factors to determine the phenotype of an organism. Those are gene, environmental factors, and gene by environmental interaction. Let us take an example. Take G1 and G2. G1 and G2, we're going to cultivate at the same location. G1 and G2 going to cultivate at the same locations, at one location. At the end of cultivation, variations are expected in their phenotype, like the yield is maybe vary, the color is expected to vary, the height and other traits are expected to vary. This is due to the variation in gene, since the location or all environmental factors are kept constant for both of the genes, for G1 and G2. So this is the variation due to genotype. On the other hand, if you cultivate G1 at different locations, maybe highland and lowland will show variation due to environmental factors, since the gene is 1, which is G1, but the variation is due to environment, since it is in different locations. And the other factor is gene by environmental interaction. For example, different gene G1 and G2 cultivated at different locations. One may be on the highland, the other may be on the lowland. At the end, there will be variations on their phenotype. This variation is due to gene type, because there are two different types of genes, and due to environment, because they're cultivated at different locations. So this is the effect of gene by environment interactions. So those are three important factors which determine or regulate the expression of one's phenotype of the organism. So what factor will influence the expression of a particular phenotype in the organism is A, environmental factors. Yes, it is a factor. B, gene. It is a gene factor. C, gene by environmental interaction. It is the factor. D, all of the above are factors to regulate the phenotype of one's organism. So D is the correct answer for question number 8. Question number 9. Guanine plus ribose sugar is what? Here, nitrogenous base with ribose sugar is a nucleoside. A nucleoside is a monomer for nucleotide. Many nucleoside plus phosphate is a nucleotide. Two or more nucleosides together is a nucleic acid. There are two naming systems for such nucleosides. That is, we have to add a suffix SIDE for purine nitrogenous bases. Purine nitrogenous base include adenine and guanine. The pyrimidine nitrogenous base consists cytosine, thymine, and uracil in case of RNA. And also, we're going to add a suffix DINA for pyrimidine nitrogenous base. Based on these naming systems, we're going to select the correct from the given alternatives. A, guanidine. No, guanine is the purine nitrogenous base, so we're going to add suffix SIDE. Guanoside, that may be correct. Guanine, it is simply a nitrogenous base, rather the nucleoside. Guanotide, no, there is not such expressions for guanine. So B is correct naming system for guanoside. This is the nucleoside naming systems. When we name the nucleotide, we have to add mono, di, or triphosphate. That is based on the number of phosphate. For example, if we add phosphate here, guanoside monophosphate, if the phosphate is 1. If phosphate is 2, guanoside monodiphosphate, and if it is 3 phosphate, guanoside triphosphate. We can naming nucleoside in such ways also. Question number 10. In the DNA double helix, which of the following is true? Our keyword is DNA double helix. It's a keyword. So therefore, which one is true for the DNA double helix? In this question, in the DNA double helix, there are nitrogenous bases as a ladder. Purine bind to pyrimidines. Purine bind to pyrimidines. That means adenine with thymine with triple hydrogen bond. Guanine binds to cytosine as the with 3 hydrogen bond. Those are the ladders of the double helix, and the sugar and the phosphate and the sugar are the side chains. In the double helix of DNA, those are happen. Therefore, choice A says sugar and nitrogenous bases make the side chains, not nitrogenous base. B, purine and pyrimidine nitrogenous bases make a ladder. That may the correct answer. C, sugar and phosphate group make the ladder, not sugar make a ladder, rather the side chain. D, phosphate group and nitrogenous bases make the side chains, not the nitrogenous base. So the correct answer is B. Nitrogenous base form the ladder, sugar and phosphate form the side chains of DNA double helix. Question number 11. From the following nucleotide sequence, how many hydrogen bonds exist? How many hydrogen bonds exist from this given sequence? In these questions, the keyword is how many hydrogen bonds exist in this sequence. In this question, we're going to remember that the presence of two hydrogen bonds between the adenine and thymine. There is two hydrogen bonds between thymine and adenine. And also there is three hydrogen bonds between guanine and cytosine. Three or triple hydrogen bond between guanine and cytosine. Guanine and adenine are the purine nitrogenous base, while thymine and uracil are the pyrimidine. Thymine and cytosine are the pyrimidine nitrogenous base. In case of RNA, there is uracil, also the pyrimidines. So the purines bind to or combine to the pyrimidine nitrogenous base in the case of double helix DNA. So in order to answer this question, simply count the number of hydrogen bonds between each combinations. Here, 80, we have 2 nitrogenous base, we have 3 nitrogenous base, we have 3 nitrogenous base, again we have 3, we have 2 between TA, 82, TA, there is 2 between GC, we have 3 between TA, we have 2. When we count all those nitrogen bonds, we will get 22. Therefore, in this sequence, there are 22 hydrogen bonds. So D is the correct answer for question number 11. Question number 12. In the double helix DNA, if cytosine accounts for 36%, then what is the proportion of adenine? Here the indicator is cytosine account 36. This is given. Cytosine is equal to 36%. Cytosine is 36%. Here adenine, with the ratio of adenine to thymine is 1. That means adenine is proportional or equal to thymine, while the ratio is 1. The ratio with guanine to cytosine is 1. That means guanine is proportional to cytosine in the double helix DNA. The other means adenine plus thymine plus guanine plus cytosine is equal to 100%. This is 100%. Here adenine plus thymine and adenine is proportional. We can say adenine plus guanine and cytosine is proportional. Cytosine also 36. We can say 36 plus 36 is equal to 100%. Here we have 2 adenine plus 72, 36 plus 36 is 72 is equal to 100%. Here 2 adenine is equal to 100 minus 72. 100 minus 72. Here 2 adenine is equal to 28%. That is 100 minus 28. So adenine is equal to 1 for 14%. So the correct answer for question number 12 is C, 40% for adenine and thymine. Question number 13. Which of the following nitrogenous bases combine with triple hydrogen bonds? The keyword is triple hydrogen bond. Which nitrogenous base form triple hydrogen bonds in the double DNA helix? In these questions, as we said before, there are two hydrogen bonds between adenine and thymine and three hydrogen bonds between guanine and cytosine. So triple hydrogen bond is formed between guanine and cytosine. Choice A, adenine and guanine, both are a purine. They cannot combine together. Choice B, thymine and cytosine. Those also the pyrimidine. They are not combined to form a ladder. Choice C, guanine plus cytosine. This may be the correct answer. D, adenine and thymine. Those form two hydrogen bonds. So choice C is correct for question number 13. Question number 14. Which of the following includes other components? In these questions, it is simply to recall the chemical composition of molecules, biomolecules. There are atoms or chemical elements which used to build up the monomer, that is the monomers or micromolecules. Those micromolecules build macromolecules like protein, lipid, nucleic acid, and carbohydrates that are the monomers. Those macromolecules are the important component of cell. From the given alternative, let me show you the composition of each. The nucleotide is composed from nitrogenous base plus sugar, ribose sugar plus phosphate. The nucleic acid is a DNA or RNA composed from the nucleotide 1, nucleotide 2, and nucleotide 3. Two or more nucleotides used to build a nucleic acid with the covalent bond called phosphodiester bond. A chromosome, a nucleic acid plus the amino acid. There is amino acid composition in the chromosomes. The nucleoside consists nitrogenous base plus the ribose sugar. Therefore, in these compositions, the one which include all are the chromosome. Chromosome is included. In the chromosome, we have nucleic acid. In the nucleic acid, we have nucleotide. In the nucleotide, we have a nucleoside. In the nucleoside, we have nitrogenous base and ribose sugar. The nitrogenous base may be purine and pyrimidine. So the correct answer is C for question number 14. Question number 15. In a plant cell, DNA can't be found in blank. In plant cell, and DNA can't be found in blank. The keyword is plant cell and DNA. Based on location, genome is found in nucleus, which is called a nuclear genome, and the genome found in cytoplasm also called cytoplasmic genome. So there are two types of genome based on location, nuclear genome and cytoplasmic genome. In the cytoplasm, there are cell organelles like chloroplast in plant, mitochondria, the Golgi complex or Golgi apparatus, the ribosomes, the vacuoles, and other cell organelles are found in the cytoplasm. From those cell organelle, chloroplast and mitochondria are self-autonomous since they have their own DNA in their own membranes. They have DNA in their own membranes. Therefore, if you're going to need extract a plant tissue, you're going to get a DNA with a nucleus, with cytoplasm, like chloroplast cell organelle and mitochondrial cell organelle. So in this question, in plant cell, DNA can't be found in blank, which cannot be found. So Golgi complex is a cell organelle but which have not its own DNA within its membranes or within its organelle membranes. So Golgi complex is the answer, the one with the absence of DNA. Question number 16 says, as per the position of centromere, chromosomes can be metacentric, submetacentric, acrocentric, etc. This is blank. In this question, we look centromere as a keyword. Centromere is a keyword. Based on this location, we can take chromosome metacentric, submetacentric, and acrocentric. What it says the question. Here, centromere is a place where two sister chromatins cross over. This is one sister chromatid, this is the other sister chromatid. Therefore, this place is the centromere. The position of centromere is different as different chromosome types. Based on this crossover, the chromosome may be metacentric if the P and Q arms are equal. That means the centromeres are at equal positions of the sister chromatid. Submetacentric if the arms are unequal. In general, it shows a physical variability of the chromosomes. It may be on the middle of the second middle or the end, and also it may be off-centric. So the position of this centromere give a different appearance of the chromosome. That appearance is the physical appearance. So the correct answer for question number 16 is the physical appearance of a chromosome. Physical appearance of chromosome. So choice B is correct for question number 16. Question number 17. The micromolecule that used to build nucleic acid is what? The keyword is micromolecule or monomer for nucleic acid. Here we have amino acid, which is the building block of protein through the connecting force of peptide bond. There is peptide bond between amino acid to build protein. The nucleoside is the building block of nucleotide with the phosphate group. Fatty acid is the building block of lipid. They are monomer for lipid. They are macromolecule for lipid through ester bond. Nucleotide is the building block for nucleic acid with the phosphodiester bond between those nucleotides. Therefore, the correct answer is D for question number 17. Question number 18. What types of bonds exist within the main strand of DNA? The keyword is types of bond. We have four major covalent bonds: the glycosidic bond, the peptide bond, the ester bond, and the phosphodiester bond. They are corresponding to the four major biomolecules. Glycosidic bond is found within carbohydrates. That is, it connects the monomer or monosaccharide to form a polysaccharide. Peptide bond is found between amino acid to form protein. So the bond within protein is peptide bond. Ester bond is found between the fatty acid, to form lipid. So ester bond is found in lipid. Phosphodiester bond is found between nucleotide to form the nucleic acid. So the bond or the covalent bond found in nucleic acid is phosphodiester bond. So the correct answer for question 18 is D. Question number 19. From what starting point can a genetic engineer or biotechnologist regenerate organism? Regenerate organism. Regeneration means the recovery of the whole organism form. We can grow the organism into its lower form. If it is plant, it should have a root system, and also it should have the shoot system. Regeneration, the conversion into the whole organism. So in this question, the keyword is to regenerate the organism. What starting point we have? This is simply to recall the hierarchy of life. In this regard, atom or chemical element are used to build molecule, a monomer and a polymer, a monomer to polymer. Those polymer or those molecules are used to compose the cell component. For example, the cell wall composed from the carbohydrate called cellulose. The cell membrane is the cell component composed from lipid and protein, that is lipoprotein. The nucleus consists chromosome. Within chromosome, there is a nucleic acid and amino acid. So those molecules used to build the cell component. If one is missing, the cell cannot be said cell. So cell is the smallest structural and functional unit of life. This tells us there is life in cell. We can culture cell to recover the whole organism. Even single cell can culture and recover the whole organism. All cells with a similar function give tissue to organ to organ system, the organ system together is the organism. We human being, the plant, the animal, and the microorganism are the organism with the complete organ system. And if one of the organ system is missing, the organism cannot say organism. Atom, question number 19, choice A, atom, not the correct answer. Molecule, not the correct answer. Cell is the correct answer. The organelle, alone or organelle cannot recover the whole organisms. If you culture only the mitochondria, you cannot recover the whole organism. So cell is the smallest and structural functional unit. So you can culture cell to recover the whole organism. The correct answer is C for question number 19. Question number 20. In the history of genetic engineering or biological science and development, which one of the following has not been addressed yet? This is to recall you or to remind you the developmental stage of science, especially genetic engineering nowadays. the development of genetic engineering is attractive in different streams of biotechnology, in agriculture, in medicine, in environment, in industry, and in other areas. It is become too attractive. So the question number 20, choice A, human cancer therapy are applied in medical biotechnology. That is correct nowadays. It is working. CRISPR trial in human. Yes, this is applied nowadays. Recombinant DNA technology and gene cloning. This is also activated. None of the above. All those applications are working nowadays through genetic engineering. So D is the correct answer. None of the above are addressed. All are working nowadays. Question number 21. Genetic engineering can be defined as, this is simply the definition of genetic engineering. Individuals or organizations may have a different definition for genetic engineering as per their aim, as per their activity. So you may define genetic engineering in different words, in different senses. So we're going to look those alternatives. The correct definition for genetic engineering is a process of gene manipulation, genetic recombination, the breakage and reunion of DNA molecules. Yes, this is the genetic engineering definition. B, genetic engineering is isolation, introduction, integration, and expression of foreign DNA into other organism. This is towards the formation of transgenic plant. That is the correct definition of genetic engineering. This is correct also. C, manipulation of existing genes with new or foreign genes isolated from other organisms. This is gene manipulation to improve its traits against the adverse environmental factors. So C also correct. D, manipulation of genetic architecture. It is genetic architecture. We change the map of a gene. We can change the appearance, the trait of an organism using recombinant DNA technology. This is also correct. So all are the correct definition of genetic engineering. So the correct answer is A for question number 21. Question number 22. In the general process of genetic engineering, which of the following will be the first task among others? In this question, the general process in the activity of genetic engineering, what sequential steps we have to do the genetic engineering of one's organism? In the process of genetic engineering, there are sequential steps. First, we're going to isolate the donor host for the gene of interest, where we gene of interest from. Second, we take a sample from that specimen and extract a tissue to get a gene of interest, that is cell extraction, tissue extraction. Now we have cell suspensions. The cell suspension undergoes cell lysis, that is a breakage of the cell wall, the cell membrane, and other organelles to expose those biomolecules. Then we're going to isolate or separate DNA from other biomolecules. We pick up DNA, rather protein, carbohydrate, or lipid. Fifth, we have to purify DNA. We have to purify DNA. Then DNA quantification and purity conformation. We confirm the quantity, we confirm the purity using the spectrophotometer, the nanodrop, or gel electrophoresis, as we have the apparatus or the facility. Then, after having this conformation, we're going to fragment a DNA using restriction enzymes. Those restriction enzymes cut a DNA into a certain piece as per the restriction site. After that, we have to prepare the vector to load this gene of interest. We have to have a vector. Then the vector going to design and construct, that is the recombinant DNA formation. Vector plus a gene of interest is the recombinant DNA. We form recombinant DNA. This recombinant DNA is loaded to transferred to the cloning host. So we have to prepare cloning host before transferring. After having cloning host, we transfer recombinant DNA into the cloning host. After transferring the recombinant DNA to cloning host, we have to prepare the selective marker. They are actually loaded with the recombinant DNA. So we have to select the transformed and untransformed cell by using those selective agents, maybe antibiotic or herbicide. Then we have to prepare the recipient host where a gene of interest transferred to gene of recipient host is important. We have to infect this recipient host with a cloning host, maybe bacteria, going to infect the host, and the genome going to fuse to the plant genome, maybe plant. Then we have to select a transformed cell using a marker in the recipient host. Then, after having those transformed cells, we have to regenerate this cell into the whole plant or the whole organism that is going to be transgenic. After regeneration, we have to scale up and commercialize the transgenic organism to the end users. So based on this general requirement or general process, from those given alternatives, which one is the first task in the process of genetic engineering? Vector design and construction. Before having a vector, we have to have a gene of interest. Regeneration. Before regeneration, there are many tasks. Transfer of recombinant DNA to cloning host. Before formation of recombinant, we need to have gene of interest. So prepare gene of interest is the first task among those given alternatives. So B is correct answer for question 22. Question 23. If someone needs to develop transgenesis through the principles of genetic engineering, what requirements will he or she need? This question, development of transgenesis, transgenic organism, that is, in principle of genetic engineering, what requirements are important or fulfill in our laboratory to develop that transgenesis? This question, to do this genetic engineering, there are requirements. These are hosts, a donor host, the cloning host, and the recipient host. At least two hosts, the donor and the recipient, and also cloning hosts are important. Host is important to get a gene and to receive a gene of interest. The other is the enzyme, maybe restriction enzyme to degrade or cut the DNA, ligate enzyme, which is to link or combine two DNA ends. Enzyme is an important gene manipulation tool. Vector, this is a vehicle or carrier of a foreign gene to the new organism, to the recipient or to the cloning host. Vector is an important requirement in the process of genetic engineering. The other is the marker gene, which is to differentiate the transformed and untransformed cell. Marker are a pattern which is to distinguish transformed and untransformed cell. So marker is an important tool in this process. The other is some laboratory skills. Cell and tissue culture skills are important to regenerate the cell into the whole organisms. Those and others are important to do genetic engineering. So from those given alternatives, a restriction and ligase enzyme, that is important too. Donor and recipient host is important. Vector is important. Efficient tissue culture skill is required, and all are important to do transgenesis in the principle of genetic engineering. So the correct answer for question number 23 is E. Is correct. Question number 24. Blank is a DNA molecule used for carrying exogenous DNA into a host organism and facilitates stable integration and replication inside the host organism. In this question, carrying exogenous DNA is an important keyword and facilitates integration and replication. And DNA molecule is also another keyword. In this question, we try to define each alternatives and we'll get the answer there. So, gene of interest, GOI, is the DNA molecule or DNA sequence, nucleotide sequence, which carry a gene of interest, that is a desired trait, the one which carry the desired traits. Vector are the vehicle or the carrier of a foreign DNA or a gene of interest into the new organism, the recipient. Promoter is a DNA molecule which is used to initiate transcription. Enhancer, a DNA molecule or nucleotide sequence itself and used to facilitate transcription. So from those alternatives, vector are a DNA molecule which is due to carry the gene of interest and facilitate the integration and replication of the gene of interest in the host organism system. So the correct answer for question 24 is B. Question number 25. Which of the following can be used as a vector? Which one of the following is a vector? In this question, the keyword is a vector. Which one is a vector? Vector is a keyword. So, vector is the DNA sequence that carry gene of interest into cloning host. Vector can be prokaryotic, example plasmid in bacteria. Vector may be viral vector, for example lambdaphage is a vector. Eukaryotic vector, for example yeast artificial chromosome is the eukaryotic vector. Vector may be hybrid vectors, for example phagemid and cosmid are the hybrid vector, they are artificial vectors. So vector can be plasmid can be a vector, virus can be a vector, artificial chromosomes can be vector, they are artificial to satisfy the need of the scientist over the limitation of natural vectors. So all those can be a vector to carry a gene as per the type of host. So the correct answer for question number 25 is D. Question number 26. Which sequential order is correct in steps of molecular cloning from left to right? In this question, sequential order of molecular cloning are the indicator for this question. They are keywords, sequential order of molecular cloning. In molecular cloning, there are sequential steps. First, isolate the donor host for gene of interest. Second, extract the tissue from the donor organism. Here we have cell suspension. A cell suspension goes to cell lysis to break the organelle and expose the biomolecules, that is DNA. Then DNA separation from other biomolecule. We have to separate DNA from other biomolecules. Fifth, DNA purification. We have to purify a DNA from other ingredients. Sixth, DNA quantification and purity confirmation using different facilities, spectrophotometer or nanodrop, as per the facilities that exist in our laboratory. Seventh, DNA fragmentation using restriction enzyme. We have to fragment a DNA within the appropriate size, actually as per the restriction site. 8. Prepare appropriate vector to carry that gene of interest, appropriate vector. After having a vector, we have to design and construct with a gene of interest, that is recombinant DNA formation, recombinant DNA. This recombinant DNA transform into the cloning host. We have to transfer recombinant DNA to cloning host. Select the transformed cell using marker, and so on. So in molecular cloning, we have those sequential steps. From the given alternatives, number 1, restriction deletion of target gene. 2, choice of donor host. 3, creation of recombinant DNA, that is formation of recombinant DNA. 4, choice of vector and restriction digestion. 5, selection of organisms with vector sequence. 6, introduction of recombinant DNA into cloning host. 7, cell lysis. All those sequential steps are disordered, so you are requested to reshape the correct order. I hope you order those sequential steps in molecular cloning and you will get the correct answer, C. I hope you got C is the correct answer. Please try it. Question number 27. What important feature should be fulfilled while choosing the vector for molecular cloning? In this question, the keywords are feature for vector in molecular cloning. Important feature of vector which used for molecular cloning. So feature of vector for molecular cloning are the keywords for these questions. When we see these questions, we remember about the important feature of vectors that used for molecular cloning or expression of a given trait. So the important feature of vectors are they should be self-autonomous. They should be self-replicating capability, self-replication. They have selectable markers. As much as possible, they are small in size with large insert size. Extra, an important feature of the vectors. In addition, based on functions, there are two types of vectors. Those are cloning vectors, which used to amplify a given gene of interest within there as a recombinant DNA. The other is the expression vector, which used to express a trait through transcription process. So those expression vectors should have a promoter, which is to initiate transcription, and terminator, and ribosome binding site. Those are an important feature or additional feature of expression vectors. So in this question, which important feature is important for molecular cloning? A, self-autonomous. Yeah, the vector should be self-autonomous in order to amplify that gene of interest. It should be self-autonomous, is correct. Replicating inside the host, it should be replicate itself to amplify that gene of interest again. So B also correct. C, must have promoter and terminator. This is that much not a feature for molecular cloning vector or cloning vectors. So this may not the answer. For the time being, we suspense this one. We suspense this one. C, D have selectable markers. Every vector should have a marker to distinguish the transformed and untransformed cells. So this is an important feature for those amplifying or cloning vectors. D, all except C. So from those given alternative, promoter and terminator is not an important feature for amplifying or molecular cloning vectors. So E, all except C, is the correct answer for question 27. Question number 28. Question number 28. This is a blank space questions. Blank is a naturally occurring extra chromosomal DNA molecule that can autonomously replicate inside bacterial cell. Here is the keyword is extra chromosomal DNA molecule. Extra chromosomal DNA molecule is the keyword. To answer this question, simply to define each alternative. Ribosome. This ribosome is the cell organelle. It is not a DNA molecule. It is simply a cell organelle, which is used for amino acid synthesis. Open genes are a DNA molecule that used to metabolize different biomolecules. Plasmid is extra chromosome within bacteria and used as a vector. Promoter is a DNA molecule or a nucleotide sequence which is used to initiate transcription. So from those alternatives, choice C or plasmid is the correct answer for question number 28 to satisfy this blank space according to the definition. Question number 29. The bacterium that confers pathogenicity on the recipient host is due to blank. In this question, the keyword is confer pathogenicity. The bacteria that confers pathogenicity. Here they are plasmid within bacteria. Plasmid is extra chromosome within bacteria. In bacteria, they are plasmid. We know plasmid in bacteria. And this plasmid give additional trait for that bacteria. There are different types of plasmids within that bacteria. For example, F plasmid, the Cole plasmid, the virulence plasmid, the degradative plasmid. All those are different types and give a different trait for that bacteria. That bacteria also act differently in different environments. So the bacteria that confers pathogenicity is due to the presence of plasmid. This type of plasmid is the virulence plasmid. This is the virulence plasmid. Note the resistance plasmid. This resistant plasmid gives resistance ability for the bacteria, for different antibiotics, for different chemicals. Note the degradative plasmid cause pathogenicity. This degradative plasmid give a threat for bacteria that degrade different matters, that decompose different materials. So the plasmid that found in bacteria enable the bacteria to cause pathogenicity over the recipient host. So D is the correct answer for question number 29. Question number 30. Why do scientists prefer to design artificial plasmid over natural plasmids? This is a reason why scientists are going to design artificial plasmid over natural plasmid. This is a reason. There are two types of plasmids. These are natural and artificial plasmid, as it is indicated on the questions. Artificial plasmid and natural plasmid. Natural plasmid may not satisfy the need of the scientist due to they may have a small or large size. Their size may be small or too large. And they may have poor marker genes to distinguish or to select transformed and untransformed cells. And they may have low replication ability. Anyway, they cannot satisfy the need of the scientist. For that reason, those scientists are going to design artificial plasmids. They already design artificial plasmid to overcome the problem of natural plasmids. Therefore, from those given alternatives, the main reason is that not for accessibility. For accessibility, it is not a matter of availability. It is not a matter of the transferring mechanism, transferring means. So it is a matter of size, a matter of feature. So C is the correct answer for question number 30. Question number 31. Which of the following can be a cloning plasmid vector? Cloning plasmid vector. Here the basic keyword is cloning, plasmid, and vector. It is a cloning vector and it is plasmid. Since there are two types of vectors, those are cloning vectors and expression vectors. Here we're going to select the cloning plasmid vectors because there are different types of vectors: plasmid, viral, artificial chromosomes, and also other hybrid vectors. So cloning and plasmid are the keywords. From this alternative, you may explore and become familiar through your reading. But from those given alternatives, the cloning plasmid will be APET. This is the vector BPUC19. Yes, this is a vector. All those are vectors, but A and B, PET and PC19 is a plasmid, because when we nomenclature or read a vector, we have P at the base and exponential is the discoverer CU and 19. CU is University of Columbia, which discovered this plasmid. Number 19 is the order of discovery. This is the 90s plasmid of that is the time of discovery. SV40, it is not a plasmid, it is a viral vector. Lambda phage, also not plasmid, it is viral vector. So the correct answer will be from A and B. Here B is the correct answer for cloning plasmid. PET is the expression vector. So B is the correct answer for question number 31. Question number 32. In this question, which of the following does not represent the hybrid vector? The keyword is does not represent the hybrid. We're going to choose the one which is not hybrid. Vector may be natural or artificial. The artificial one is hybrid. So the hybrid vector is developed by combining the best features of different vectors. So these alternatives, lambda phage, it is not the hybrid, so will be the correct answer. So we will suspend this one. Cosmid is the hybrid vector of the virus plus plasmid. Important feature from virus and important feature from plasmid. Phosmid, again the virus plus plasmid. Phagemid, also another viral vector plus plasmid. All those important features of the virus are taken, and the important feature of the plasmid also taken and combined together give those hybrid vectors. So the correct answer for question number 32 is A. The lambda phage is not a hybrid vector. Question number 33. The process of transferring DNA into the eukaryotic host cell is called blank. What it is called? The process of transferring DNA into eukaryotic. Here is DNA transferring into eukaryotic cell or host cell is the keyword. DNA or eukaryotic cells have organelle membranes. Eukaryotic cells are organelle membranes. In order to make opening through the membrane, that is the way to make opening, infection has to take place due to the presence of vir genes, that is the virulence genes. These virulence genes infect the organelle membranes in order to make opening and fuse into the nuclear genome or cytoplasmic genome. So in eukaryotic, infection is allowed through the virulence genes rather than that vector. So from those given alternatives, the gene transferring process in eukaryotic host cell is the transfection. Without transfection, there is no infection. Without that infection, there is no any opening. Without opening, the DNA have no chance to fuse from the recipient genome. So infection is take place. So C is the correct answer for question number 33. Question number 34. Blank is a vector into which the desired DNA fragment has been inserted to enable its cloning in an appropriate host. This is a blank space, but we have keywords a vector and desired DNA fragment as insert. DNA fragment, that is a gene of interest. Here we have plasmid, that is a vector. We have vectors within this blue, green colors, and a gene of interest with this purple. There is an DNA fragment as insert. So this DNA sequence consists two different sequences from two different sources. One from plasmid or from that vector, and the other a gene of interest. So it is the combination of those two DNA sequences or nucleotide sequences. So which one is the appropriate description of this statement? A, restriction enzymes. No, not the restriction enzymes. Those enzymes are used to cut or degrade the DNA fragment into different pieces as per the restriction sites. B, recombinant DNA. Recombinant DNA is the combination of two or more nucleotides. This may be the correct answer for this question. Bacteria is the host. D, cloning host. This is the host. Bacteria is the host. Cloning host also similar with choice C. So the correct phrase or word which is suitable to complete this blank space is B, recombinant DNA molecule, which consists the nucleotide sequence of a vector plus the sequence of the gene of interest. So B is correct answer for question number 34.
Question number 35. Which enzyme is a popular tool for molecular cloning and is also called molecular scissors or scalpels? In these questions, the enzyme and molecular scissors are indicators or the keywords for the questions. Which enzyme is used as a molecular scissors? In this question, we remember that there are different types of enzymes. For example, there are restriction enzymes, which are used to restrict or cut a DNA fragment in different pieces. And there are also ligate enzymes, which are used to join or link DNA fragments. There are modifying enzymes. There are synthesizing enzymes, and we're going to use those enzymes as per our needs in molecular cloning. Therefore, in order to answer this question, we have to look at the choice. Which enzyme is used as a molecular scissors during molecular cloning? A, ligase enzyme. Ligase enzymes are those which is to ligate or link to DNA fragments. They are used to bind to DNA fragments. B, end modifier enzymes. Those enzymes are used to modify the end of a DNA fragment for various purposes. C, degradative enzymes are those which are used to cut and degrade a DNA fragment at the middle or at the end of the DNA, internally or externally, that means. D, polymerase enzymes. Those are enzymes which are used to synthesize the DNA fragments or used to extend the length of a DNA, which is to synthesize new DNA templates. So the correct answer for question number 35 is C. Degradative enzymes are the molecular scissors, which is used to cut a DNA at different positions, maybe internally or externally of that strand. Question number 36. Consider yourself in the third person involved in the development of the restriction enzyme from bacteria Bacillus subtilis, subspecies Murberg. How should this enzyme be named according to the international naming conventions? This is simply the naming system of bacteria, the enzyme from a certain bacteria. Naming system of the enzyme which extracted from the bacteria. Bacillus subtilis, subspecies Murberg. Subspecies means the strain of that bacteria. Therefore, to name restriction enzymes, we have a certain orders. First, take the capital letters of the genus name. Capital letters of the genus name. The genus name is Bacillus. So we have to take B, capital letter B. Second, take the first two small letters of the species name. The species is subtilis. So we take the second two small letters of the species name, S and U. S and U. Third, take the first letter of the strains. Here is the strain Murberg. So we take the first letter of the strain name. The strain name. Fourth, add the Roman number, which indicate the order of discovery. That is the third person. That is the third time to discover that restriction enzyme from the bacteria, from this given bacteria. So this is the third discovery. We write the Roman number in such ways. So the name of this bacteria is BSUM3. So choose the correct answer with this naming system. So BASU, it is not correct. No, it is not correct. BS, no, it is not correct. So C is correct. This is not correct. C is correct for this naming system.
Question number 37. The types of DNA fragment indices that are formed when the restriction endonuclease, like EcoR1, cleaves the DNA at different locations, generating fragments with protruding terminals are blank. This question is somewhat long statement, but we're going to select the keywords. The keywords are types of DNA fragment index. DNA fragment index is the keyword. That are formed when the restriction endonuclease, like EcoR1, cleaves at the DNA different locations. Yeah, the second keyword is at different locations. At different locations. That is a DNA fragment index that have a different index, means a different that cut at different locations. Protruding terminal. Protruding terminal is also another keyword. This is an important phrase. DNA fragment index at different locations and protruding terminals. Those are indicators for the answers. Protruding terminals, unequal ends of that DNA. There are two restriction patterns of DNA due to the action of restriction enzymes. Those are blunt end and sticky ends. Blunt ends are happened when restriction enzyme cuts the DNA at the same locations of both DNA strands at the same locations. Means if we have this DNA strand and the restriction enzyme cut at the same point, and this is a blunt end. And sticky ends are happened when the restriction enzyme cuts those DNA strands at various locations. If we have this strand, one cut at this point, the other at this point, and the second strand also at this point and this point at a different locations, and resulted an equal end of DNA fragments. This is the sticky ends. So from those given alternatives, which one is the correct description of this statement? A, blunt end. No, it is equal. It has equal ends. B, mutilated ends. No, there is not such type of patterns. C, sticky ends. This is the correct answer. D, single-stranded ends. This is no such type. There is no such type of patterns. So sticky ends are the unequal DNA fragment ends due to the action of restriction enzyme. So C is the correct answer for question number 37. Question number 39. Question number 39. 38, sorry. Question number 38. Which enzyme is used to digest the hairy loop formed in the synthesis of double-stranded cDNA? Which enzyme is used to digest the hairy loop? The hairy loop that formed during the synthesis of double-stranded cDNA. This cDNA is complementary to messenger RNA. So which enzyme is responsible to cut this hairy loop, this curve? This is the cDNA at this condition have such type of pattern. This pattern. This is used as one strand, and this may use as the second strand. So how to cut this hairy loop? Which enzyme is cut this hairy loop? This hairy loop. This is the action of exogenous enzyme, actually, exogenous enzymes, but which exogenous enzyme or which exonuclease is important. A is exonuclease. This is general. There are different type of exonuclease enzymes. So this is more general. This is more general. DNase, this is DNA based and used to degrade the whole DNA strand. This is degradative, so it doesn't cut the loop only. S1 nuclease, this is exonuclease enzyme, which used to cut such type of looping during cDNA synthesis. So this is maybe the answer. We suspend this one. RNA polymerase is the enzyme. This is the RNA-based enzyme. Therefore, it is not for to cut this such looping. So the correct answer is C for question number 38. Question number 39. DNA ligase enzyme is applicable to blank. This is simply to know the application of ligase enzyme. This enzyme is used to join or link to DNA fragment. It may be DNA fragment, or it may be gene of interest with the vector and with the various application also. So read the alternatives and choose the one which is the application of DNA ligase enzyme. So application of ligase enzyme. A ligate vector and inserts DNA in recombinant DNA. In the recombinant DNA, ligase enzyme is important. So choice A is correct. If used to seals the nicks of double-stranded DNA, if in the different applications or in different manipulations, the DNA may be damaged, may be broken. In order to repair, in order to seal that nick or damaged, DNA ligase enzyme is important, is to seal, to repair that nick. So this is also another application of ligase enzyme. The ligase enzyme also important to join linkers with a DNA fragment. Linkers are a nucleotide sequence. DNA fragments are also another sequence. In order to join those sequences, DNA ligase is important. So all those are the application of the ligase enzymes. So D is the correct answer for question number 31. Question number 40. The synthesis enzyme that used to synthesize cDNA by using messenger RNA as a template is blank. The key word of phrase is synthesizing enzyme. Synthesize enzyme of a cDNA from messenger RNA. That enzyme is RNA-based enzyme, which is used to synthesize cDNA, complementary DNA synthesis. So from those given alternatives, which one is suitable for this statement? A, DNA polymerase. DNA polymerase actually is a synthesizing enzyme, but it is DNA-based, not messenger RNA-based enzyme. So it is not suitable for this question. B, RNA polymerase. This is also synthesizing enzyme. During the transcription of DNA into RNA, it is used to break the hydrogen bond between those DNA double helix. So this is haven't a role in cDNA synthesis, that is on transcription of DNA to messenger RNA. Reverse transcriptase. Basically, cDNA is synthesized through reverse transcription. So the enzyme responsible to synthesize cDNA strand from messenger RNA as a complementary strand is reverse transcriptase. So this is synthesizing enzyme for this cDNA. D-terminal transferase is another synthesizing enzyme, but this is used to synthesize or modify the end or the terminals of that strand. So not suitable for this question. Therefore, C is the correct answer for question number 40.
Question number 41. After the formation of one strand of cDNA, what enzyme will you use to remove the messenger RNA template? In this question, it is cDNA, enzyme, and messenger RNA. After the synthesis of the first strand of cDNA from messenger RNA, which type of enzyme is responsible to remove messenger RNA from that combinations? Enzymes have different roles when we do the gene library, that is cDNA library. To form cDNA, we have sequential activities. First, we have to isolate messenger RNA. Second, we have to synthesize the first and second strand of the DNA, that is cDNA. While doing those, we use enzymes to extend and to remove those strands from that combinations. So the enzyme which is responsible to remove messenger RNA is S1 nuclease. No, S1 nuclease is the exogenous enzyme which is used to cut the loop while we synthesize the second strand of cDNA. It cut at this point. P ribonuclease or RNase. This enzyme is RNA-based enzyme which is used to degrade the RNA. It may be the answer for the questions. C. DNA polymerase 1. This is synthesizing enzyme which is a DNA-based, which is used to synthesize DNA strand. So it is not to remove messenger RNA. RNase, this is also DNA based, which is to remove a DNA. When we add RNase in these solutions, it degrades cDNA too. So it is not the correct answer for question 41. So choice B is correct for question number 41. Question 42. Blank is short, chemically synthesized, self-complementary, double-stranded oligonucleotide, which contains one or more restriction endonuclease sites with both blunt ends. Here, short, chemically synthesized DNA sequence, self-complementary, double-stranded with multiple endonuclease sites, restriction endonuclease sites. It is multiple restriction sites and have both blunt ends. Which alternative is suitable for this definition? Therefore, you have to be familiar with those alternatives. What is adapter? What is linker? What is gene of interest? What is recombinant DNA? So through your reading, you become too familiar with those words. For the time being, linker is a suitable definition for this statement. So the correct answer is B for question number 42.
Thank you for watching, and go to the next video number, which is video number 3.